Hello, and welcome back to biology. Today we want to focus on a couple of those macromolecules that we've talked about before. That would be the nucleic acids, the DNA, RNA, and the proteins. So basically what we want to talk about today is how do you go from DNA to proteins? What does that mean? What does it entail? What is DNA really, and what are the proteins doing? Although we did already talk about the functions of proteins a little bit. Okay, so we're going to start off with a question. It became apparent to Watson and Crick after completion of their model that the DNA molecule could carry a vast amount of hereditary information in its sequence of bases, phosphate sugar backbone, functional groups, complementary pairing of bases, ability to separate and replicate. So here's the thing. The answer is going to be the sequence of the bases. That code is what's going to determine what kind of protein you get. Its ability to separate and replicate is really important, and what we're about to talk about now, because that ability to separate and replicate is what allows things to actually grow and develop and all that good stuff, and other things too, which we'll talk about later. But it's the bases, the sequence of the bases, that really matters in terms of what it's saying. So we want to talk about the first thing, replication, okay? Replication is really important because what happens is you get an identical, hopefully, copy of the DNA in a new cell, right? And so it can copy itself and then you can have a new cell and that can copy itself, and this faithful copying, this copying it identically, is really the key to having multicellular organisms. Now, the process is what's called semi-conservative, and that means that one strand, right, is going to, when you have the replication process, one strand is going to be the parent strand and one strand is going to be the older strand, and I will talk more about that in just a minute. But next, right, here's your question. During the process of replication, which bonds in the diagram of DNA below is or are broken? All of them? Here's 1, here's 2, here's 3, here's 4, here's 5, here's 6, and here's 7. Well, this one actually has 2 answers. It's going to be these hydrogen bonds in between the base pairs that are going to be broken. And so it works a little something like this. You have your parent molecule. The strands separate where these hydrogen bonds are, and then as the strands separate, there's a whole mechanism, right, of proteins that come and they form this replication complex, and they are going to allow for these sides to be open. Well, there is a nucleotide base that wants to have a bond. These nucleotide bases are just kind of floating around, and so they just fall in line as the attractions are stronger when they're closer. And so you get this lovely new strand. And so you have 2 identical daughter molecules, one parent strand, one new strand together. Okay, and so what that means is that when you look at this, one strand is going to be the template strand and one strand is going to be the coding strand. Okay, now this is a lot more complicated than we're actually going to talk about, but I wanted to put this in there so you'd see it, right? So on the leading strand, okay, so we're going to be doing replication. It's from the 5' to the 3' end, and that just tells you the orientation of these strands, okay? And we're not actually going to talk about that a whole lot, but I just want to point it out. So that means that this strand right here is the template strand, and this strand will be identical to the coding strand, which is down here. Now, this strand that's on our coding strand for this will be the template strand for this strand, which will match this parent strand up here. So that's the coding strand for this one. Okay. Now, an interesting thing, the DNA is only going to grow at the 3', it's only going to elongate at the 3' end. And so here you have your DNA polymerase, which is the complex that's going to make that happen. And then you have some other things over here. You have some stabilizing proteins, you have some helicases, which actually unwind the DNA from that lovely spiral that we know about. And then there's some RNA primers and things like that that are also important. DNA ligase is going to show up. And we're not actually, again, going to talk about all of these things. I just want you to know the overall process. But I do want to say something really interesting about this other strand. This other strand is oriented backwards to this one, which means the 3' end is over here. So the replication is going to be at the replication fork, but it's going to have to go backwards. And so this strand, this what's called the lagging strand, is actually replicated in a series of short fragments called Okazaki fragments. And when it's all said and done, it'll be just like this one. It's just that the process is actually really cool. So, moving on. So if cytosine makes up 20% of the nucleotides in a sample of DNA from an organism, then adenine would make up what percent of the bases? So let's think about this. We know that cytosine is going to bond always with guanine, right? So if 20% of the strand is cytosine, that means that 20% of the strand must be guanine, which is 40% total. So the other 60% is going to be adenine and thymine. So half of that 60% will be adenine, and half of that 60% will be thymine. So 30% is the answer. Okay, now, once we have our DNA replicated, we can do all sorts of things like cell replication. It's very exciting. But what is the point of having the DNA to begin with? And the point is that there are sections of the DNA which are called genes, and those are going to be transcribed into RNA, and then the RNA is translated into proteins, and the proteins are the things that are going to do the work in the cell. Okay, and so this is kind of an overview. So let's go through this. Okay, so here you have your strand of DNA. Now you have another complex that's called the RNA polymerase, and this is going to be the thing that transcribes the DNA into RNA. So in replication, you had DNA polymerase because it was making DNA. In transcription, you have RNA polymerase because it's making RNA. And so same kind of idea, you unwind the DNA, and then one side is transcribed into RNA. And so it's the template strand here. So this strand is going to match in sequence this strand. So this strand is the actual gene. This is the template for the gene. Okay, and so it grows the same way, 3', 5' to 3'. And so it's just going to go until it reaches the stop sequence, and then it's going to break off and go off and do its thing. Now, RNA is actually really, really cool. And RNA is probably the first of the genetic material, right? Because RNA, and then you get DNA later on, but there's a hypothesis, or theory actually, it's actually a theory, called the RNA world. RNA is capable of being an enzyme. It's capable of catalyzing reactions and things like that. And so it, in some ways, is like a protein, but it carries the message like DNA. And so probably it's the first thing to evolve, and then DNA shows up later, and then RNA takes all these other forms. So, different types of RNA actually have different types of function, and there are lots of different kinds of RNA. We're going to deal with 3 of them. The mRNA, which is the messenger RNA that codes the protein. rRNA is the ribosomal RNA, and it actually makes the copy machine, or not the copy machine, but the translation machine that's going to make the amino acid, like the protein, the polypeptide strand. Then there's tRNA, which is transfer RNA, and that is what's going to bring the amino acids to make that protein to the ribosome. Now, I put this in here so you could see that there's actually lots of different kinds of RNA. Now, what we're going to be interested in most is the mRNA, because this is the instruction RNA, right? It's going to say what kind of protein you're going to have. Now, it is actually read in 3 sets of 3 base pairs. Those are called codons, right? There are actually 64 possible codons because there are 4 nucleotide bases. Remember, in RNA, it's going to be C, G, A, and U instead of C, G, A, T, right? But they're still read in 3. There's 64. There's only 20 amino acids. This is really exciting, actually, because that gives you redundancy. You can have multiple codons for the same amino acid, and that gives you some flexibility. There are, however, some special codons, and those are the AUG, right, the methionine, which is always the start. That tells the ribosome this is where you are going to start making the protein, right? And then there are 3 stop codons. This is where the ribosome will detach and the protein is finished. Okay, so which of the statements below is false? The genetic code is universal. Degenerate codons specify the same amino acids. The genetic code is overlapping, and the genetic code is triplet. Well, it is universal, right? There are 4 DNA nucleotides, and there are 4 RNA nucleotides, and actually there are really only 5 nucleotides total if you think about it. Degenerate, that means that you have this redundancy, that multiple codons can code for the same amino acid, and we'll talk about why that's important in just a second. The genetic code is triplet. It is read in those triple codon or triple base pairs, which are the codons. It does, however, not overlap, right? There's no overlap. You run through, and the codons are going to be every 3 bases. Okay. So here is just a amino acid chart, right? A coding chart. And you can see that you have your 20, and here, like, these all code for arginine, right? So do these. So you have 6 possibilities, right? These 4 all code for glycine, right? You have 4 possibilities for that. 6 for leucine, 3 for isoleucine, right? And so you see where I'm going with this, yeah? There's methionine, that's the only one of its kind, and then here are the stop codons, right? There are 3 of those. Okay, so a geneticist isolates a gene and the corresponding mRNA for a specific trait under study. She finds that the mRNA contains 1,000 fewer bases than the DNA sequence. Did she isolate the wrong DNA? Because that's weird. I mean, if you're copying, you should have, if you're transcribing, you should have a match, right? So yes, the mRNA should contain more bases than the DNA sequence because bases flanking the genes are also transcribed. No, the mRNA was partially degraded after it was transcribed. Yes, mRNA is made from a DNA template and should be the same length as the gene sequence. And no, the final mRNA contains only exons. The introns were removed. Well, the answer is 4. So what does that mean? Okay, so when you look at this picture, you can see here's your gene, right? It's transcribed into pre-mRNA, right? Precursor mRNA. Only part of this in eukaryotes is going to be useful in terms of genes, and those areas are called the exons. In between the exons, you have introns. This can be transfer RNA, it can be ribosomal RNA, it can be junk DNA that's not going to be turned into anything, okay? But whatever happens, in order to get mature mRNA, right, the one that's going to be translated, you have to cut out those introns. And that leads to a very interesting thing that can happen. You can change the gene based on the way you splice it. And splice it is cutting pieces out. You're always going to cut out the introns, and then they'll go off and do whatever they do, okay? Some of them just degrade, some of them are other types of RNA. But where you cut it can make a huge difference as to what you get. In this case, exon 3 is actually excised. It's cut out. So you only have 1, 2, 4, and 5. That makes 1 gene. Here, you're going to lose 4, right? And so this is a different mRNA. It's going to make a different protein. And that's actually really cool, and it allows for a lot of flexibility. So if you have, say, 20,000 genes, right, in the human genotype, you have somewhere between 100 and 150,000 different kinds of proteins. Well, that means that those genes are being cut in different ways, and it's called alternative splicing, and it's very cool. Okay, now, once you get your mRNA, it's going to leave the nucleus, and it's going to be translated, and it will be translated into a protein, and this is kind of just a really basic overview of what's happening. This is the transfer RNA, right? You're going to have a ribosome, this is the mRNA, and these are the amino acids. Okay, so the transfer RNA is this nice little folded piece, and if we were to kind of lay it out, it would look something like this. You'd have these little loops. Occasionally you'll have this little 4th loop right here. But the important parts that you need to know are the acceptor arm right here. This is it's right here in this picture. This is the amino acid attachment site. Each transfer RNA matches a specific amino acid, and it has an anticodon here. This anticodon is going to line up with the codon on the mRNA. Okay? So, as, right, the mRNA is going to move through the ribosome, which I'm going to talk about in a second, then you're going to have this hooked up. And I'm going to show you that in a picture just momentarily. But before we do that, I want you to show you the ribosome. The ribosome is the building machine. It's the actual, it's a machine. It's a biological machine. It puts the amino acid chain together, and in reality it looks like this. That's all RNA and proteins. It's very, very cool. But basically you have the mRNA go through the ribosome here. You can see it's going to lay across here, and there are 3 openings in that ribosome. Now, this is much more complex than we're actually going to be talking about, but I want to show you what it looks like. Okay. Now, for a more simpler version, so here's the ribosome. There are those 3 slots have letters. There's E, the exit, P, the polypeptide, and A, the acceptance site. Okay, so the transfer RNA is going to come into the acceptance site. It's going to match the anticodon here is going to match the codon here. It will have an amino acid at that acceptor arm, right? And then once it comes in, then there will be a polypeptide bond that forms as they move through. So first one is going to be methionine. F-methionine is for prokaryotes like bacteria, okay? We're going to be using MET, not F-MET, just as an aside, because we're going to be dealing with eukaryotes. But anyway, so here's the point. This guy, the methionine, would have come into the acceptor site. The ribosome will move down, making the next codon open. Then the next transfer RNA with whatever this matches will come in. And then when this transfer RNA shifts over, a polypeptide bond is formed between these 2 amino acids. Then it shifts over again, and this transfer RNA here will end up in the exit site. It is released out of the exit site, and it goes off to pick up another amino acid. The one that was in the acceptance site then moves into the polypeptide site, right? Then you get a new one in the acceptance site, right, the acceptor site, and then a new bond is formed, okay? Oh, sorry, I went the wrong way. Okay, so we're going to go this way. So the bond is formed, then everything shifts over, and then this guy is going to leave. So this is where we're picking up. So this guy is leaving, and then this guy is going to come in. It's going to form the polypeptide bond, and then the ribosome is going to shift 3 nucleotide bases over, and this guy is going to get kicked out of the exit site. And so you're just going to keep going until you hit the stop codon, and once we hit the stop codon, it just kicks right off, right? The ribosome releases the mRNA and goes off to do another one, okay? This is just another version of it. Here you've got the A site and the P site. This one actually doesn't show the E site. It just shifts over and pops out. But this just gives you, you know, this gives you the names. Like here's the codon recognition. Then there's the peptide bond formation. Then there's the translocation, which is the moving of the ribosome down the mRNA, and then, you know, it goes away. Okay. So an antibiotic interferes with the ability of a ribosome to move. What effect would exposure to this chemical have on a bacterial or any cell, actually? Protein synthesis would be affected. The protein synthesized will be shorter than normal. The protein synthesized will be longer than normal, or no proteins will be produced. Well, the answer is actually no proteins will be produced, because if the ribosome can't move, it can't shift to the next codon, the new transfer RNA can't move in, the polypeptide bond can't be formed, and then it can't shift again, right? It's got to move down the mRNA to form the amino acid chain. Okay, so, right, what happens if there's a mistake? So you have this DNA, and sometimes there will be changes to the DNA itself for various reasons, right? Maybe there's a mistake in the copying. Maybe it's been exposed to a mutagen, which is a chemical that will trigger changes to DNA, and there are lots of different kinds of mutagens, right? And so we're not really going to talk about those. Or you can be exposed to harmful radiation, which is called ionizing radiation, and that will break DNA bonds and form dimers and all kinds of things. Again, we're not going to talk about the specifics, but it causes damage. So you get mutations, right? And there are different kinds of mutations. So this is just a little table of the point mutations, and we're going to talk about each of these. There's a substitution, right? There are frame shifts because of insertions or deletions, okay? And then we're going to talk about some more in a second, but let me show you how they work, okay? So let's say that you have normal DNA. Yay, you get a normal polypeptide. Woohoo! Now, if you have a silent mutation, you have something that changes. So here you have the AAG, right, instead of AAA. And so you've got this change. Well, in a silent mutation, remember there are multiple codons that can code for the same amino acid. In this case, the mutation doesn't make any difference at all to the final amino acid sequence, even though that it's changed, okay? A missense mutation means that you do change the amino acid, and so you get a slightly different amino acid sequence. Now, sometimes that's not a problem, because some amino acids behave the same way as others. Sometimes it's a real big problem because the amino acids behave differently, and then you can't have the proper folding, or it doesn't have the proper active sites or whatever, and that's a real problem. Okay. Now, a nonsense mutation means that when you change this, right, one of these, it actually changes to a stop codon, which means that you have this totally mutated mRNA that's cut short and will not do what it needs to do. Okay, so those are the three substitutions, right? Now, the two different types of frame shift, you have an insertion, right, which is where you can add in a whole nucleotide base. That is going to shift the line so you get entirely new amino acids, or you can take one out. Again, it's going to shift the line, but this way, and you're going to get entirely new amino acids, right, from that point on. The thing about frame shifts is you can't actually tell if it's an insertion or deletion, unless you actually know and can compare and all this good stuff, but otherwise it's very hard to tell. Okay, so, right, next thing, gene versus chromosome mutations. Chromosome mutations involve the whole structure, right, of the chromosome, whereas the mutations we've been talking about are just involving a change in a nucleotide, just the base sequence of the gene. Chromosome mutations, right, are a different kind, but basically what you have is you have your gene, and then it is duplicated, and you should get an exact copy of the chromosome, right? And a chromosome is just a long continuous strand of DNA and some of the proteins and some RNA and all this good stuff. But anyway, they're hooked together at a centromere, and we'll talk about these more a little bit later on, but just to give you an idea, the short arm is the P arm, the long arm is the Q arm. So this is P, this is Q. Here's the centromere. Your heterochromatin is the DNA is coiled really, really tightly, and it is not transcribed well. The euchromatin, this unwinds easily, and this is typically where your genes are going to be. The ends of the chromosome are called telomeres, and we'll talk about this later on when we talk about aging a little bit. And then this, of course, is a real chromosome, right? Again, we'll talk about this more when we talk about cells. Here's a karyotype, which actually shows all of the chromosomes in a human female. See that little XX? Now, here are the changes in the chromosome structure. First, you can have a deletion. That means there's a break in the chromosome, and part of it pops out and is deleted, and then you get a shorter piece here. Maybe you cut in the middle of a gene, maybe some genes are missing. This is really not a good thing. You can also have an inversion, where a part pops out and it flips around and then reattaches, so now it's in the wrong order. This can be a real problem. It can also not be a problem at all. It just depends. And then you have translocation. That is where a piece of one chromosome reattaches to a different chromosome. A non-homologous means that it is not a copy of the same one. It's an actual different chromosome. So if we just pop back really quickly to the karyotype. Let me get back there. Okay, it's like, so maybe this one pops off and is added to this one, or they switch arms there or something like that. So you're combining two different ones. Okay, we saw all of that, yay. Come on now. All right, so here's your question. Which mutation would most likely be lethal? All mutations are lethal, all mutations are beneficial. Chromosomal mutations, gene mutations, or any mutation has the possibility of being lethal. Well, actually, any mutation can have the possibility of being lethal. If it screws up the function of a protein that's important, it's going to be a problem, right? Okay, so molecular clocks. We can actually date separation of species using this RNA, okay? RNA, because it has to make the ribosome, is conserved, which means that it changes very slowly, because changes in those sequences that code for the ribosomal RNA, if that doesn't form right, you don't get a ribosome, and you can't actually you can't actually translate the mRNA, because the ribosome doesn't form. So this is a really good way to map how species have evolved over time, by looking at these tiny changes in RNA. Now, mitochondrial DNA, which we're going to talk about mitochondria later on, this is the DNA that's not in the nucleus. it can change really quickly. And so you can use this to explore changes in species over several thousands of years, right? And that's actually how we know about relationships among humans, for instance. But I just wanted to show you that we can use these things to make observations about the relationships among organisms. And because it actually works really, really well, we've changed the classification system to be based on ribosomal RNA sequences. And so it used to be just kingdoms. The domains that we now learn, the eukarya, bacteria, and archaea, are going to be based on these ribosomal sequences, right? And so that's pretty cool. And then here is just, these are the bacteria, the archaea. These are extremophiles. We're almost never going to talk about those, but they're really cool. They live in really inhospitable, what we consider to be inhospitable places. And then here are the kingdoms in the domain eukarya. And these are all the eukaryotes, and we'll talk about those as we go through. All right, so that was just a little bit about classification. We'll do that more later. If you have any questions, please let me know in class, and I will see you next time. Thanks for listening.